Hello, I'd like to welcome everyone to today's webinar on breeding for nutrition in organic seed systems. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. You can find all of our published articles, videos, and our many recorded webinars on organic farming topics on our website at extension.org/organic_production. This webinar is a rebroadcast of a presentation given at the Organic Seed Growers Conference in Port Townsend, Washington this past January. There are several live broadcasts that we recorded at the conference that can be found at extension.org/pages/61925, but I'll be typing that link into your chat box in just a moment so you know where to find those presentations. Before we begin, I'd like to pass this over to Micaela Colley, who will be moderating the presentation, and she's going to introduce the speakers.
Good morning, everyone. This is Micaela from the Organic Seed Alliance. We have three presenters this morning, the first of which is Philipp Simon. He's an USDA Agricultural Research Service geneticist and a professor of horticulture at the University of Wisconsin-Madison. His research on carrot and garlic genetics and breeding is conducted to improve these crops for growers and consumers. Some of his key areas of interest include carrot and garlic genetics and the development of genomic tools and genetic improvement of carrot root knot nematode resistance, crop diversity and origins, and the nutritional quality and flavor of both carrots and garlic. Philipp is also the PI on a new project called the Carrot Improvement for Organic Agriculture. Alice will give you the URL for that website at the end of this talk. Jim Myers will be the second speaker. Jim Myers holds the Baggett Fraser Endowed Chair of Vegetable Breeding and Genetics in the Department of Horticulture at Oregon State University. He works on a number of crops, including dry and snap beans, edible potted pea, broccoli, tomato, winter and summer squash, and sweet corn. His main interest has been to improve vegetable varieties for disease resistance and human nutrition, while maintaining quality and productivity in improved varieties. Jim is also breeding tomatoes, broccoli, and summer squash for organic systems. His latest variety release is the high anthocyanin tomato Indigo Rose. The third presenter is Walter Goldstein. Walter Goldstein grew up and received his education in Washington State, but now lives in Wisconsin. He has bred corn under organic conditions since 1989. He was the research director at Michael Field's Agricultural Institute for 25 years. He has recently begun a research and education organization called the Mandaamin Institute for breeding nutritionally valuable crops and promoting healthy farming practices.
All right. Yeah, this is Walter Goldstein. I'm executive director and research director of Mandaamin Institute in southeastern Wisconsin. And I'd like to talk to you today about some of the experience that we've had with breeding corn for nutritional value for organic farmers. Our particular emphasis has been on protein, carotenoids, and taste, and we've worked on it about 20 years or so, and I'd like to say a few things about what we've learned. In corn in general, there's been a tremendous increase in yield over time, but it's been accompanied by a decrease in grain quality in terms of nutritional value. In particular, breeding corn for yield has led to a progressive increase in the starch yield per acre, but what we're seeing is a decrease in the percentages of the protein content in the grain. And the protein content and the quality of the corn are, of course, extremely important for the country in terms of feeding livestock. Corn generally makes up more than half the feed for livestock, and its nutritional value is very important. That's also important for organic farmers. Organic protein is very expensive, and of course it takes protein to raise animals and to build body and make eggs and produce milk and so on. We've had listening sessions with farmers starting back in the early 90s where we've asked organic farmers what kind of corn they actually wanted to have. And it became quite clear that amongst many of those, particularly those who were raising their own livestock, that they were very interested in the nutritional value of corn. and they were actually very aware that corn nutritional value had decreased over time in terms of protein content. They also were interested in mineral content, vitamin content, and they were also concerned about taste. Just to give a little bit of orientation as to what kind of decline may have occurred over time, we recently analyzed a set of Native American corns from the Plant Introduction Station in Ames, Iowa. They had been grown there, and we analyzed their protein, oil, starch, and also the content of the essential amino acids lysine and methionine in the grain of those varieties. And we had, you can see we had 14 Hopi flour corns, 4 mixed flour and flinty corns. We had Hopi Mexican June derivatives. We had 11 varieties derived from other tribes. And we also, just for check, we put in values that we had for some of the Corn Belt dents. Nowadays, it's very common that we're running somewhere between 7.5%, 8% protein on our hybrid corn varieties, and you can see the enormous difference that we were seeing between that and the varieties, the Native American varieties. Now, these weren't grown side by side. Nevertheless, we do have this kind of experience when growing some of these older varieties relative to Corn Belt dent varieties over time, that this is a kind of, we can see similar differences to this, though not always so extreme. Also, when we look at the lysine and methionine content, we see that, as particularly with these flour corns, that they're very high in lysine and methionine. Now, lysine and methionine are very important because they're essential amino acids, and they, if we go back, we can see that that we're practically having not only about double the protein content, but almost double the essential amino acid content in these flour corns relative to Corn Belt dent. And this sort of gives you an idea that there may have been a decline in terms of nutrient density, actually a very strong decline, even though, even while we celebrate the fantastic improvement of corn for its yielding capacity on a per acre basis. The Native American corn isn't the only corn that we may look at in terms of being a treasure chest for nutrients and nutrient density. If we go to the Amazon and look at some of the multi-alurone corn, we can see that it has very high levels of minerals, protein, potentially more B vitamins, and also phytosteroids. And this has to do with the fact that it has not only a high protein content and a high essential amino acid content, but also because it has multiple layers of the alurone. The alurone is this little layer of tissue beneath the pericarp, the outside sort of skin of the seed. And in the case of these Amazonian corns, they can have up to 7 layers relative to normal corn, which only has 1. Let's go back to methionine and lysine. Why did we focus on them? As you probably know, proteins are made up of amino acids, and some of those amino acids they're called essential because you can't make them in your body. You have to get them from your nutrition. And methionine and lysine are generally regarded as being the primary limiting amino acids for humans, for hogs, for poultry, and for dairy cattle. Especially with poultry, methionine is crucial. It's very important for the overall health of the birds. It's important for egg production. Lysine is the second, regarded as being the second most important. And corn makes up more than half of the ingredients of poultry food in the Midwest and further east, but it's naturally low in the sulfur-containing amino acids methionine and cysteine, and also it's low in lysine. And we make up for that by combining corn with soybean meal, and also, even with organic farmers, by supplementing with a synthetic methionine. Now, this has been very controversial. and yet it's been deemed as being necessary. Organic egg production has quadrupled since 2003, and even though organic farmers probably aren't interested in feeding synthetics to their birds, neither do they want to have nutrient deficiencies. And so nutrient deficiencies and synthetic methionine, or both of them, are probably not consistent with organic ideals, but nevertheless, there seems to have been a kind of partnership there. However, by 2015, it should be phased out, and so there's a need for high methionine feed, high methionine corn, in order to replace it. And we do have corn that is very high in methionine. In order to be able to breed for it, we needed to have a quick test, and we utilized a near-infrared spectroscopy for measuring quickly methionine and lysine. And so what we found was that we could breed high methionine and lysine corn in grain that was hard, had a hard endosperm, and in grain that was soft. Now, the hard endosperm types differed in that if we're going to have a hard endosperm type that was high in methionine, it by necessity had to be very high in protein. And unfortunately, in breeding with hard endosperm types, it's difficult to get from farm to farm to get a reliable protein content in the grain. It's somewhat similar to growing hard red winter wheat, that sometimes you get the high protein and sometimes you don't. And so that makes it a little bit more problematic. But the soft endosperm types are a little bit more stable in terms of maintaining a high percentage of lysine and methionine in the protein. They're not as closely tied to the protein content of the grain. We have been working with partners who've done feeding trials with broilers and layers, and that was Organic Valley, and also with the University of Minnesota. And our trials showed that our high methionine corn, soft endosperm, could replace synthetic methionine without losses. On working with those soft types of corn, we discovered that, well, the one that we first worked with, which possessed the flowery 2 gene, was associated with a lower seed weight and a lower yield. But we have found soft types that don't seem to have that problem, and we've continued to work with them. Now, in general, breeding with high methionine, breeding high methionine and high lysine corns, they are higher in protein content, and we expect there will be a yield penalty. The farmers are not going to get the same yields. But we do also expect that when we're utilizing the best of our cultivars, we're going to be having more protein being produced per acre. I'll show you some high performance liquid chromatography data here. This is the wet chemistry method that's generally used to analyze grain, and here you can see normal corn. These actually is the average of 1903 values for corn. This is done over many years. Actually, the protein content of corn has sunk over time. I think 9.5% it would be considered to be high protein now. We're seeing, as I said, generally between 7.5% and 8% protein in normal corn now. And we're generally seeing, instead of 0.21%, we're seeing about 0.17, 0.18% methionine. Now, contrast that with the hard kernel methionine corn, 28 different varieties, and you can see that the protein content difference and the difference in methionine. And look at here at the flowery 2 corn, and you can see the difference in protein content with the normal corn, and also the difference in the methionine content and in the lysine content. There's a strong increase for both of those associated with working with this type of corn. When we've taken this corn out onto farms and we've tested it on large-scale conditions, we aren't achieving the same protein levels as we're achieving on our test plots. They're lower, And on average of 3 different farms where we were testing the soft kernel type, we had only 10% protein. It was still high by the standards of normal corn, but not as high as what we'd seen in our test plots. However, with this flowery corn, it's still maintaining a rather high level of methionine and lysine, and so that's the benefit of using these soft kernel types. Now, going back, you'll recall that I said we developed a quick test that's a near-infrared spectroscopic calibration, and it's non-destructive. We pour the seed through a machine that throws light at the seed. It bounces off, the spectra are changed, and patterns in the changes of the spectra have been found empirically to correlate with the essential amino acids of methionine and lysine and cysteine. And so we've been looking at these 3 essential amino acids and doing these calibrations. This was work that was done at Iowa State University at the Grain Quality Lab with Charlie Herberg and his group, and with funding from the state of Wisconsin and other places, USDA. And what we discovered was that we had broken the essential relationship between protein and essential amino acids that has bedeviled other attempts in the past to derive these sort of correlations. The problem is this, is that generally the essential amino acids are components in protein. And when you do this kind of calibration, you end up just estimating the essential amino acid content based on the protein content of the grain. It's just simply a mathematical relationship. However, you can see what's happened here is that if we look at the correlation between the protein content and the actual values for lysine and methionine, you can see that the relationships are rather low. However, what we've been able to achieve with the calibration are rather high correlations or predicted values, which means that we've instead of simply recalculating essential amino acid content from protein, we actually are predicting, this method is actually predicting the essential amino acids. So we've actually made a rather large breakthrough by being able to do that. Now, looking at our hard endosperm types, which are these little red triangles, this is a set of S3 lines grown on 2 different sites. And you can see here what we're looking at is the percentage of protein in the grain of those S3 lines and the percentage of methionine in the grain. And this is for a particular population. What we did here was we developed hard endosperm high methionine varieties and soft endosperm high methionine varieties out of the same population. And you can see the advantage that we have here, that we have a higher methionine content per amount of protein with that soft endosperm type. So that's leading us, of course, to be going in the direction of working more with the soft endosperm types. Here's a little bit on the trial that was done with broilers at Organic Valley and layers at the University of Minnesota. The palatability of the soft kernel cultivars in both set of trials was very high. They actually had to restrict feed of the soft endosperm types to avoid feeding frenzies. We are hoping in the future that we will be able to do larger trials. There's a team called the Methionine Task Force made up of a set of about a dozen organic poultry companies who have a keen interest in alternatives to synthetic methionine. Now, I mentioned earlier that as we go into quality in corn and we're looking at more protein, that we may lose our yield. And this is, of course, a big concern. How can we manage to combine quality and yield? And the high methionine corns generally are high protein corns. And if we select simply for high protein, we can actually be selecting for seed that's smaller. It actually has reduced endosperm and seed size. So it's important not to do that. We want to be able to select corn that produces high protein without having a reduction in that endosperm size. So we actually need to be, at least indirectly, targeting alterations in the nitrogen physiology of the plant so that it's more efficient at utilizing nitrogen and getting it from soil organic matter and putting it into the seed. In terms of the way that we're selecting now, we've shifted going in the direction of selecting for a high concentration of methionine and lysine in the grain, but also with looking for yields per acre of protein so that we can combine the two things and not lose yield as we're selecting for quality. How are we doing? Well, in 2009, we took a set of our new hard endosperm hybrids, and we tested them out on 9 organic and 9 conventional sites. And that was done together with the U.S. Testing Network, which is a group of smaller companies, and also with USDA. If we looked at what kind of yields we got on the organic sites, our hybrids yielded 87%. Of what the other conventionally developed hybrids were yielding. On the conventional sites, lower, 81%. And the best yielding high methionine hybrids on all organic sites were HM11 and HM2, which yielded 94% and 91% as high as the average for all the elite hybrids that we tested. It's fairly typical, and this seems to happen very often, is that the best high methionine hybrids on the conventional sites are not the same thing as the ones that yield well on the organic sites. They were HM1 and HM6, and they yielded 88% as high as the average elite hybrids tested. And in terms of the agronomic characteristics, they were very similar to the other hybrids in terms of lodging, etc. Now, we also looked at quality, and to evaluate differences in grain quality, on one site we self-pollinated ears, and we analyzed them for quality. And the high methionine cultivars averaged 12.9% protein and 2.8% methionine on a total dry basis, and that's pretty typical for what we've seen in the past. That's about 43% more protein and methionine than is generally found in normal corn plants, in corn hybrids, in this study and in our studies on previous years. And if we projected yields of protein, we were producing approximately 1/3 more protein and methionine per acre than did the conventional hybrids, but about 13% less starch. So in terms of looking into the future and feeding the world, that makes sense. Why don't we go in the direction of breeding for nutrients per acre rather than just breeding for weight per acre? Phil addressed that a little earlier on, and maybe we can talk about that more in our discussion. Phil also talked about carotenoids, and I would like to point out that corn is a very valuable source of carotenoids. He mentioned that also. But one of the ways in which we get carotenoids is through eggs. When we feed chickens when we feed chickens corn, the carotenoids are accumulated in the yolk. And it turns out that the yolks of eggs are perhaps one of the best ways of getting carotenoids into the human body. Apparently the cholesterol in the egg is very important in terms of actually getting the carotenoids into the bloodstream of people. And we can really do something here with respect to turning yolks even oranger. And in fact, on some of our high carotenoid corns that we feed on my own farm, we end up with yolks that are sometimes looking as orange as pumpkins. And if you look at some of the retinoid active carotenoids in the grain of the corn, we look over here on the far right. We see a Northrop King, a NC Plus check, which was a decent, well-colored hybrid, and we compare it with some of the populations that we have been selecting for carotenoid content. You can see that it is possible simply by selecting for visual orange, bright orange, and bright yellow to be able to increase the carotenoids in the grain. Now finally, I'd like to touch on the issue of taste, which has been mentioned also by my fellow contributors here. However high it yields, somebody has to eat it. And they're only going to eat so much, and it may not taste as good as it should. And it also, not only is it a question of nutrition, it's also a question of enjoyment of life. I'm showing you here a picture of some cattle that were eating one of our corns. The farmer greatly appreciated this corn. It was derived from native corn, and the farmer believed that his cattle were extremely greedy at eating this particular variety versus the hybrids that he also grew. They would eat everything from this corn, whereas they would leave the stalks and a lot of the leaves of the other varieties. So there's often these sorts of factors that we don't consider when we do our breeding, but actually really make a difference for the end user. You can see those greedy little eyes. Now, we've tried to get to the issue of taste in a simple way, in a screening procedure, and this was our best attempt to do it. What we did was we took our breeding lines and we would make little cakes from them, and we'd replicate 2 small cakes made very simply from a mixture of corn flour, water, salt, and baking soda. We had to be very careful with the salt in terms of the amount that we put in there because we could really bias things. And what we would do would we would line up all of our colleagues, and every day they would take a little slice out of those cakes, and they would judge them in a very simple 1 to 5 scaling system. And it turned out over time, as we looked at the scores that we were getting, was that they actually were very highly correlated between the different people who were scoring, which was a surprise to me. In general, the result was that the more modern hybrids, the more crossing that was done with conventional inbreds, the poorer the taste. So we had very potentially lost something in terms of taste by breeding without considering it and focusing on yield.